Well, I'm coming from Vietnam. It is actually one of the most plastic polluted country. So I understand really well the negative effect of plastic pollutions into the environment and also in our daily life.
Doctoral student An Nguyen at the Center for Young Sin Bioscientists is using the tools of synthetic biology to develop new, cost-effective pathways to produce sustainable biomaterials.
My research is focusing on producing chitin and chitosan from a microbial source. The main aiming of my research is to lower the production cost of these biomaterials. And we are focusing on two solutions. The first one is to engineering the yeast and the fungi so it can actually producing more chitin and chitosans. By producing more chitin and chitosan in one batch, it's lowering the production cost. And the second approach that we are focusing on is co-producing this biomaterial with other valuable compounds such as lipid, enzyme, or biofuels.
Chitin is an excellent structural component that can be widely used in materials, food, pharmaceuticals, and even in energy production. Currently, the main source of chitin is crustacean waste in the fishing industry. However, obtaining chitin from animals and plants is not sustainable in the long run. For example, shrimp and oyster farming are known to cause detrimental damage to natural marine systems. The solution would be to grow the chitin using biotechnology. An is using microbial fungi to produce this highly versatile polymer in bioreactors. Often, when microbes are cultured, the desired end product is secreted either into the growth medium or inside the cells, and the end product needs to be separated. But in this case, the entire cell biomass can be utilized.
Because chitin and chitosan is also the component of the cell wall of yeast and fungi, we can actually utilizing this biomass waste to extract this biopolymer.
An is boosting the natural production of chitin by creating stress for the fungi.
The novelty of my research, at least my technology, I believe, is that I'm focusing on I'm utilizing a defense mechanism of the yeast, which gonna be activated when the yeast are in a stressful environment. What it does is that the pathway gonna increase the synthesis of the cell wall of the yeast. A simple explanation, it could be that when you are in the cold weather, you tend to wear a thicker coat. It's the same thing for the yeast. It builds a thicker cell wall.
Understanding and developing the microbial pathways benefits the whole field of biotechnology.
I'm intrigued by the complexity in such a tiny thing, with all the pathway, all the mechanism, they work in harmony with each other. Well, I'm still learning to do research, but I believe that in the future we can produce basically every consumable product from the microbial source, and it's a much more efficient and environmental friendly way anyway.